All right, hello. This is the laboratory introduction for our plant responses lab. So this is just some reminders before I get into what our lab is going to be about, is that our blog post is due this Friday at 12 p.m. And so you should be uploading this on bblearn. There is a blog post set up there, or a blog set up there. There is instructions on how to submit the blog post in the blog post folder on bblearn. We also have uploaded the rubric for you, so what we're going to be grading you based on. So take a look at that. And if you have any questions, please feel free to reach out to your lab TA. And so next week, the stomata lab report is also due. So next week in lab, and we are doing angiosperm diversity next week, and that is our last week. One thing that I will point out is we have a lot of points that are due soon. So please make sure you get those in, or those assignments in, so we can grade them and give the points to you. All right, but this week is plant responses. And so prior to coming to lab, we are asking you to read in your textbook 40.2 and 40.4 to 40.6. And so this is the only place that you're going to be getting information on plant responses. So it's important that you read the background information in the textbook first. I'll go over just a few of the basic things, but it's important to read that because it'll help you a lot. We'll also have textbooks in lab for you, and you will also be keeping this packet so you can always go back and add details in later. But with that, I'll just tell you what lab today is going to look like. So we are going to be understanding the importance of phototropism and gravitropism in plants. So how plants respond to light and gravity specifically. We will also be looking at, or trying to understand, the role of the root cap, excuse me. So you'll keep this lab packet. We're basically setting up a series of experiments that you will look at next week, right before you dive into the angiosperm diversity week, or sorry, angiosperm diversity lab. So you'll look at your experiments, complete it, turn it in. You will be graded for or from the sketches, labels, hypotheses, and mainly the detailed responses and discussions that you give. So use your textbook, ask your lab TAs what specifically you want, but I'll try to also lay it out in this intro video. All right, so with that, I'm just going to jump straight into the first thing that you'll be looking at. Well, it's not the first thing. It is one of the experiments that you'll be looking at, and so that is phototropism. So how plants can respond to light, or growth in response to light. And this can be negative or positive. And so if you think about it in the way that shoots are positive because they're always growing towards light, and roots are negative because they're always growing away from light. In this picture here, you can see we're looking at the shoot of an oxalis plant. And so this is basically what we're going to be observing in the classroom. And so what we're observing is the shoots growing up and then bending towards the light to try to get more light for photosynthesis. And so what you need to know is how plants do this. How do they bend and grow towards light? And the idea about that, or the reason, I would say, is I'll direct you down to this diagram on the bottom. And so auxin is a plant hormone, and so you'll read about that in the chapters in the book as well, but we are looking or the way that plants respond to light is by changing concentrations of the plant hormone auxin. And so when a light source changes, and so you can see in this diagram, the sun is moving to the right of the picture, and with phototropism, auxin accumulates on the dark side of the plant. And so as you can see, as it moves, the auxin, which are these little pink dots, accumulates on the dark side of the plant. And that auxin will promote cellular elongation along the dark side. So all of these cells right here elongate, and that elongation causes the curving or bending towards the light. And so that is just a pretty general description about how phototropism works. I have a video here that we can watch, hopefully. Of just some plants that are it's a time lapse of some, I guess, radish plants. And as you can see, oh goodness. Okay. As you can see, the plants that are receiving the most light are up along the front, and so they're not, they're growing pretty straight up and down, but the plants in the back here that aren't receiving as much light, they are elongating along that dark side and trying to curve towards the light. Great. Oops. Okay, so that is phototropism. And then we're going to go into gravitropism. So this will be basically like the first two experiments that you do. I'm just going to hide myself while I go over this slide. But gravitropism is the ability to move in response to gravity. And so this can also be negative or positive. So it is the opposite here in comparison to the phototropism. So shoots are negative because they're always growing against gravity, right? Because they're always trying to grow up. And roots are positive because they're always growing with gravity. So they're always growing down. And so the way that plants do this, and so right now I have a picture of a plant that's tilted on its side, and the shoots have turned and started to grow upwards, whereas the roots have turned and started to grow back downwards. And so this is one of the experiments that we'll be looking at, and you'll be focusing on how that happens in the stem system of the shoots, or in the shoot system, excuse me. So in the shoot system, auxin is also responsible for this. Excuse me. Auxin is also responsible for gravitropism. However, it is initiated by gravity instead of light. And so cells in the root cap respond to gravity and initiate this response to gravity, or this movement in response to gravity. And so in the stems particularly, cellular elongation happens where auxin builds up. And so auxin starts to build up on basically the side, the lower side of the stem. The lower side, that's so where the auxin builds up is where cellular elongation happens, which is opposite in the way that roots happen. And so auxin will also settle on the lower side of the roots, but so that auxin buildup will promote cellular elongation on the opposite side of the auxin buildup. And so that is something to note, is that gravitropism acts differently in roots and shoots. And I believe I just have a video. Let's just see if it's still there. I have a video. Sorry. I have a video of beans growing. And so this is just showing you how the root cap basically senses gravity. And so through this time lapse, sorry about the sound. In this time lapse, you can see that root cap push its way through the dirt. And so there are also special cells within that root cap that detect gravity. And so in the third experiment that you will be doing is we're going to be, sorry, I'll go back. We're going to be cutting off parts of the root cap to see what happens and how and if the roots can sense gravity after you mess with the root cap. Okay, so that is the basis of what we're going, or that's, I guess, the information that you'll need before going into lab today. There are going to be 4 parts to this. And so in part A, you'll just be observing some plants. So you'll just have to draw them and explain why you're observing what you're observing. In part B and part C, you're going to be setting up like a little experiments. I'll explain that in a second. But I wanted to say in part D is where we're going to be looking at phototropism, and you'll also just be observing plants. And so B and C is the only real ones that you'll have to wait until next week to finish before handing it in. So part B, we're going to be looking at the gravitropism in roots. And so we're going to be setting up an experiment with corn. Please also read through the directions before you come to lab of the lab handout, because it'll explain what we're doing. But basically we're going to have Petri dishes in which we're placing in an vertical position, and we're going to be placing corn seeds in different positions, like on this diagram here, and we're going to see how and in what direction the roots go in for this experiment. And I believe the next experiment you'll have to set up some hypotheses, so feel free to set up those before you come into lab. You will have to have your TA check off your hypotheses. We just want to make sure you get the points and are thinking about the experiment correctly. So yeah, so you'll set that up, leave it, come back in a week, check it out. And the part C is the role of the root tip. And so for this one, we're going to be setting up an experiment with bead seedlings. And so you'll be, like I said, removing parts of the root tip. It's important to do this under the dissecting microscope because you can't see the root cap unless you're under some sort of magnification, and we don't want to cut off that apical meris or the meristem within the root. So look to your TAs for help with that if you need. You may work in pairs for this, but I think that is it. Cool. Well, have a good lab. See you next week.